get some insight into cognitive disorders and vice versa. Okay, now I got to get back into computer mode. All right. So this is, as a card-carrying neuroscientist, like to show always a picture from Ramon Cajal. And this was taken probably about 100 years ago, where he was documenting all of the cell types in the mammalian nervous system. And clicking forward. Fast forward to just a couple of years ago, large-scale atlas efforts have been trying to document cell types. And I don't think Cajal could have envisioned quite the number of neurons or number of cells that one has been able to uncover based on transcriptomics in the brain, thousands of cells, at least in the mouse brain. Okay, so just because not everyone here is a neuroscientist, I'll just give you some numbers. In the human brain, there are roughly, depending on how you count it, 170 billion cells. That's on par with the number of stars in the Milky Way. So that's a lot of cells to be trying to understand, say, at any sort of level. And Cajal and others have documented cell types based on morphology. More recent modern approaches were documenting cell types based on connectivity. And my lab and others are using the transcriptome, trying to understand how every cell in the body can have the same genome, yet how that genome is turned on and off can lead to the diversity of cell types that we can see in the mammalian brain. And again, for the non-neuroscientists in the audience, it's not just about neurons. There are other cells in the brain, such as you may have heard of astrocytes or oligodendrocytes, but there are also many subtypes of each one of these. And this is just an illustration of the many different types of cells that one could find, for example, in the brain. Okay, so today I'm going to share with you 2 vignettes of how we can understand brain function, evolution, and disease at the cell type level using these new approaches. The first will be some published work, evolutionary approach comparing human to non-human primates. And the second will include some more recent work where we've integrated genomics with human brain measures of activity, and then our unpublished data using ex vivo slice cultures. So here's a human brain superimposed next to that of our closest genetic relative, chimpanzee, and you can see, other than the change in size, they look relatively similar. So we're trying to understand the molecular mechanisms that may underlie these distinct phenotypes that emerge at the organismal level. And this work has been inspired by the work of King and Wilson, who, when they were studying proteins of humans and chimpanzees 50 years ago now, in 1975, made this statement: their macromolecules are so alike that regulatory mutations may account for their biological differences. So this opened up the door to try and understand how gene expression and gene regulation may underlie evolution that in the brain and then perhaps the organismal behavior. And so for a lot of our comparative work, we're comparing humans and chimpanzees, but if we only compare those two, we couldn't determine on which lineage the change may be occurring. So we're typically including as an outgroup, in this case, rhesus macaque tissue, because we can obtain that more easily. And so what we do is we look for things that are different between humans and chimpanzees, but are similar between chimpanzees and rhesus macaque. And then in this case, we can assign a change specifically or assume or make the prediction that it may be changing on the human lineage. And I'm going to first start by showing you some work that is using postmortem tissues taken from the dorsolateral prefrontal cortex, also known as Brodmann Area 46. So this study was published a number of years ago now by 3 talented postdocs in the lab at the time. And we had postmortem tissue from human donors, both unaffected individuals and those who had been diagnosed with schizophrenia during their lifetime, chimpanzee and rhesus macaque. And this predates the ability to isolate single cells using microfluidics approaches that I'll get to later. And so in order to narrow down to cell types, because previously we had done this using bulk pieces of tissue, microarray bulk RNA-seq, we really wanted to see were there any changes being driven by at least large classes of cell types. So we could isolate, or the 3 postdocs could isolate nuclei, incubate with an antibody, and then put those through a flow cytometer to isolate either the oligodendrocyte lineage using olig2 as an antibody, or the neuronal lineage using new N as an antibody, purify the RNA, then carry out RNA sequencing. And this principal component analysis just showed us, either for the neurons or the oligodendrocytes, that we could recapitulate phylogenetic differences. But what we were really trying to understand was, what are the things that are changing specifically on the human lineage? And so we wanted to undertake a differential expression analysis. And when we did that, we noticed we could identify genes changing specifically in humans in neurons or oligodendrocytes. And those lists of genes are interesting. But what we noticed was, when we took into account the evolutionary distance, so the number of million years separating these species, we found that there was an acceleration, if you will, specifically on the oligodendrocyte lineage. This was surprising to us because from our prior work and the work of others, we had all noted genes changing specifically in humans in neurons. And so my initial instinct as the PI was that there was some technical issue. We had done something wrong. Why weren't we seeing more changes in neurons than oligodendrocytes? That's what the literature supported. And so what we did was, we took all of the prior study, or not all of them, but a number of the more recent prior studies, and we overlapped our new data onto those previously published data. And as I would have hoped, the neuronal genes that were changing specifically in humans did overlap with those prior studies. So what we found in the neurons was recapitulated. That was great. But again, we saw this lack of overlap. These new data changing specifically in oligodendrocytes didn't overlap with any of the previous data, suggesting it was a new result. And again, I remained skeptical of this. Surely there must be something that maybe we were doing wrong with this new approach, new in our lab. And so what we did was, we took these four prior studies, and right at this time when we were struggling with what's going on here, a couple of data sets came out using true single-cell approaches. These were from the Allen Brain Institute in Seattle. We could take that kind of ground truth data set, this initial ground truth data set, and we could use a process of deconvolution, with many of you in the room may be familiar with it. And from deconvolute our cell type fans bulk data using that single cell data, and see what it was enriched for. And what we found was that all of the prior 4 studies were indeed enriched in signal for excitatory neurons. We now know 5 years later that, using many single cell data sets and generating our own, that neurons express more genes and at higher levels. So if you take a piece of brain tissue, cortical brain tissue in particular, gray matter, and you dissociate it, or if you don't dissociate and you take it in bulk, the signal is driven by the neurons. Even if there are more non-neurons in there, because neurons express more genes, basically that is what you will observe. We could also deconvolute our fans data and show quite nicely that the new N data was driven by neuronal signals, in this case, both excitatory and inhibitory, and the OLIG2 signal was driven by oligodendrocytes. So that was great. That was a sanity check. So we had discovered something new, not because the biology was new, but just because the technology had evolved for us to be able to observe it, and observe it at a higher resolution. So that was very exciting. Okay, so now we had these genes that were changing specifically in neurons or oligodendrocytes in the human lineage, and we wanted to understand, well, what was cool about them? What are the priorities? If you were going to go back into the lab and study one or more of these genes, how could you possibly select them? And so it's fun to be back at UCLA and show this slide, which is using WGCNA, or weighted gene co-expression network analysis, which was developed by Steve Horvath here now, probably a couple decades ago. So we've used this approach over the years to try and make sense of our large-scale results, prioritize genes. And when we applied that to this data set, we were able to identify 2 human-specific neuronal modules and 2 human-specific oligodendrocyte modules. So these would be the genes that we'd kind of, or the sets of genes that we would go after. And if we visualize them, and here all of the red circles indicate a gene that was significantly changing specifically in humans, run a gene ontology, you find things that resonate with neuroscientists: vesicle-mediated transport, synapses. So if you look at the genes in neurons, you see genes that are important for neuronal function. So that's great. That's a good positive check. but perhaps not as exciting and new. In contrast, when we did the same thing and looked at one of the human-specific oligodendrocyte modules, what we found was an enrichment of genes that are involved in RNA splicing and processing. And this is very exciting to us because oligodendrocytes are an understudied, or have been an understudied cell type in the neuroscience field, although cell types go in and out of fashion in neuroscience. And my understanding is that RNA processing and splicing has not really been looked at in oligodendrocytes, but there are plenty of RNAs that are expressed throughout oligodendrocytes, and there may be some sort of trafficking that's important for how they interact with neurons. So these are entry points into studying that in some sort of cellular system, which I will kind of allude to at the end of this first half. So at the beginning, I said that we're interested in this interplay of evolution and disease. So I just wanted to remind you of that. So there are two ways that we could look at that in this particular data set. The first was to intersect our signatures of human-specific expression with publicly available GWAS data set, so genome-wide association studies. When we did that with the human-specific neuronal modules, to our surprise, we didn't find anything above this red line. We didn't find anything significantly enriched among the many at the time available brain GWAS data sets. Now, with newer data sets, this may change. No one in the lab has gone back and looked at that. However, interestingly enough, the oligodendrocytes again kind of rose to the occasion such that the human-specific oligodendrocyte modules were enriched for many brain-specific GWAS signatures, such as attention deficit hyperactivity disorder, some schizophrenia here, etc. These are non-brain GWAS, BMI, diabetes, not enriched, so very brain-specific. So that was significant and suggested that perhaps oligodendrocytes do indeed, human oligodendrocytes have some role in brain disorders. But if you are paying attention a few slides back, I did mention that not only did we have postmortem tissue from control individuals, we also had it from individuals with schizophrenia. And we carried out the same FANS approach to isolate nuclei, carried out differential expression between control and schizophrenia. And we could superimpose those differentially expressed genes with the evolutionary relevant ones. And what we found, again, neurons really did not rise to the occasion. There was no significant interaction or overlap here with the schizophrenia differentially expressed genes and the human-specific neuronal genes. In contrast, again, we found this really interesting result with the genes that were significantly downregulated in schizophrenia in oligodendrocytes overlapped with the genes that were significantly upregulated specifically on the human lineage, and vice versa, suggesting this kind of trade-off of evolutionary adaptation or acceleration and disease susceptibility, which kind of fit with our kind of initial hypothesis based on work from Tim Crow and others. So this was very exciting. And at the time, based on technology, we were really limited to neurons, oligodendrocytes. We couldn't get any more refined than that. So this was right. Again, the timing is everything sometimes in science. Single cell, 10x genomics, DROP-seq, all of these high throughput microfluidics approaches were becoming commercially available or affordable to the average lab. And a paper came out from Philip Kaidovich's group that had done single cell RNA sequencing in 4 species in this case. And they were able to make this human specificity signature of differential expression. They were using the anterior cingulate cortex. It's another part of the neocortex similar to the one that we use that's involved in cognitive function, theory of mind, frequently affected in a number of cognitive disorders. And what they found and what struck us as very exciting was that the cell types that were the most enriched for this human specificity signature were all non-neurons. So here we have oligodendrocytes and oligodendrocyte precursor cells. So this dovetailed really nicely with our finding. They went from 2 cell types to 6 major cell types. So we thought, well, there's still room for further refinement here. There's also a lot of parts of the brain that haven't been explored. neuroscientists like to focus on the frontal part of the brain. Let's look elsewhere. So I had recruited into my lab a talented graduate student, Emery, who is now a postdoc at Harvard Medical School. And we decided to look in the posterior cingulate cortex for a number of reasons. One is there's clear anatomical homology among the species. Sometimes when you get into areas of the neocortex and you have human and your outgroups, you're not really comparing the same, we're not sure if we're comparing the same brain region. This area has also been implicated in higher order cognition. It has been shown in imaging studies to be altered in schizophrenia. And most importantly, from postmortem samples, it was readily available from our collaborators. The frontal cortex has been oversampled from the non-human primate tissues in the freezer, but posterior cingulate cortex, we could get as much of it as we needed. Okay, so this was published now a couple of years ago, but here's the take-home message, was that Emery found when he looked in these tissue samples across the species and was we looked at all the cell types, but I think we were primed to look at oligodendrocytes from the previous study. He noticed something right away when he made, here, you can see many different, depending on how, if you're a lumper or splitter, many different cell types on the oligodendrocyte lineage. This blue indicates, this is our proportions, number of nuclei isolated. Oh, and I didn't say this because it's postmortem tissue, it's all nuclei, not cells. Increased proportion in the human samples in the immature oligodendrocytes. So these are the cells that can be differentiated into a mature oligodendrocyte. And when you plotted and do the statistics, what he found was that indeed there was a significant increase in the fraction of immature, so the OPC, the progenitors, and a consequent decrease in the proportion of the mature oligodendrocytes. So kind of this trade-off. We not only had the single nucleus RNA-seq, we also had ATAC-seq from these samples. It wasn't true multiome at that time. It was done independently. And using both data sets, we could find this shift in proportion. We were really excited by this, and we thought this is really surprising because it hadn't been reported in the literature from decades of folks looking at tissue samples across primates. We're also, again, skeptical and mildly concerned because, as many of you in the room may know, to generate these data sets, you take a piece of intact tissue, you dissociate it into nuclei or cells, and you're losing the three-dimensional structure. So depending on how you dissect that tissue, how you sort or obtain your 10,000 per sample nuclei, you may be biasing your results in some way. So we wanted to see if we could independently confirm and validate this finding using intact tissue. And so we did single molecule FISH, RNA scope, using markers for immature oligodendrocytes, in this case PDGFRA, or mature oligodendrocytes, MOG, across new samples, numerous individuals of new samples from the posterior cingulate, quantified that signal, and basically found the same result, an increase in immature oligos, decrease in mature oligos in the human tissue samples. So this is exciting. We could confirm our result in the posterior cingulate cortex. But as I had already mentioned, why was the posterior cingulate cortex available to us? Well, neuroscientists weren't really looking at this brain region. And so maybe there was just something unusual about it, and maybe there was a reason why a lot of people didn't care to look at it. Could we confirm, was this result transferable to other parts of the brain, other parts of the cortex? And so in order to do that, we took this published data set from the Kaitovich group and performed the same analysis. Now, you may ask, well, why didn't they find the same thing? You said this was a new result. Well, Emery at the same time had a second paper published in Neuron where he had found that folks who were dissociating brain tissue weren't taking into account pervasive ambient RNA. Remember, neurons express more genes and at higher levels. So when you dissociate the tissue, all of those beautiful RNAs that get transported to synapses spill out into your 10x milieu and get captured into droplets. And you think, oh, I found this novel cell type. Well, really, it's just ambient RNA. So what Emery did was he cleaned up the Kaitovich data set and then was able to see in the anterior cingulate cortex essentially the same result, this increase of immature oligos, decrease of mature oligodendrocytes. We also obtained additional tissue samples from this brain region from additional donors, did the SM FISH, and could quantify this in intact tissue, this increased immature oligodendrocytes. So we were super excited. We're like, wow, we have this novel result. We see it in both the posterior cingulate cortex as well as the anterior cingulate cortex. Maybe it's just something about the cingulate cortex. Maybe there's something has evolved funky in this part of the brain. What if we looked outside of those regions to other cortical areas? So we were able to look in two other cortical areas, the dorsolateral prefrontal cortex and the primary motor cortex. This was us basically deconvoluting our prior FANS data. We couldn't do exactly the same analysis. We could just look at the ratio of the two from deconvolved data. And we were also able to add in some data from Marmoset. This was a study from the Allen Brain Institute, found the same thing. Increased immature, decreased mature oligodendrocytes everywhere we looked in the neocortex. So very exciting. What if we look outside of the cortex into other parts of the brain? It's a big brain. So we were using what was publicly available to us. These experiments are expensive. There was, at the time, available data from caudate nucleus and dentate gyrus. We did not see the same result. So to summarize what we found this shift of OPC to mature oligodendrocytes in at least 4 unique neocortical areas and not in 2 non-neocortical areas. So this is very exciting to the lab. This has pushed us in all sorts of new directions, but it did also open up one really important question, like why? Why would the human brain have evolved to have this change in this ratio? It seems like there's a lot of similarities across the mammalian brain, but why this particular shift? And again, for the non-neuroscientists in the audience, this is kind of the general trajectory from a precursor to a more committed oligodendrocyte precursor cell to an immature oligodendrocyte to mature oligodendrocyte, which could be involved in myelinating axons of neurons. So this is what we kind of always think about when we think about oligodendrocytes, but if you can see all these little red squares or whatever, circles, this is from a review now 15 years ago where they're indicating all of these neurotransmitter transporters and receptors, right? So oligodendrocytes are not just in the mammalian brain in order to facilitate myelination. They're also communicating with neurons. There's also recent evidence that they can prune synapses. So there's a lot more that they can do. And so we came up with this hypothesis that I'll show you. We're thinking about testing that perhaps this increased pool of immature oligodendrocytes in the human brain has evolved to be sitting there waiting for some sort of activity signals that the neurons are sending out. So when there's experience, there's a faster response. And that's how, as humans, we're able to, no offense to Jane Goodall, able to study the chimpanzees and evolve cognitive functions in which we're, sending astronauts up into space. So able to evolve the functions that our brain can carry out. So this is our hypothesis, that this pool of immature oligos are sitting there, or not sitting there, but they're available in order to respond to whatever signals are in their environment, whether it's from neurons or from other cell types. And there's a whole literature now, as I've already alluded to, with synapse pruning, but also the role of OPCs in plasticity and injury, demyelination, and interaction with immune cells. So another interesting aspect of thinking about oligodendrocytes and human specificity that our lab is investigating. As far as we know, we're the only species that naturally develops multiple sclerosis, which is a demyelination disorder. It also has an immune component, but we have to modify model systems in order to have them have a similar disease-like phenotype. So there's something unique about the human brain that has made us susceptible to this disorder. And we think that the proportional change that we found might be at least one aspect that underlies susceptibility to that. So now that we have this result and we're pretty confident and we have this hypothesis, well, what can we do with it? Well, one thing that we want to know is how prevalent is it to other brain regions and how does it change over development? So the samples that went into the study that Emory did were all middle-aged samples, 40 to 60-year-old. postmortem tissues. So did that evolve over development? Was it like that from early age compared to non-human primate? That's something that we don't know. We know there's a differential trajectory of oligodendrocyte differentiation, specifically in different parts of the brain, namely gray matter, where we took the cortex, where there's a lot of neurons, versus white matter, which is primarily composed of oligodendrocytes. So one way we're doing this is we were fortunate enough to become part of an NIH-funded Brain Initiative Cell Atlas network. It's an atlas where they're trying to identify all of the cell types of human, mouse, and rhesus macaque and marmoset as model systems throughout the entire lifespan, from fetal embryonic through old age. And so as part of that, we're looking at 8 different white matter regions and functionally relevant gray matter over the lifespan, and carrying out multi-omic single-cell RNA-seq and ATAC-seq to see how this human-specific trajectory changes across species and across brain regions and across development. So there are multiple axes there. It's quite an undertaking, and we're about maybe 50% done with just generating the data. So I don't have results to show you other than so far the data are really high quality, and they'll be interleaved with data from other brain regions that the consortium is putting together and then made publicly available, actually, so as an atlas, which is very exciting. So this is just like an example result from a white matter sample using snRNA-seq. And I just, again, if anyone is interested in neuroscience, I just show this as a contrast to what you typically think of, and to remind you that the white matter is primarily composed of oligodendrocytes. Here are OPCs, the precursors, and then a whole bunch of microglia, some astrocytes, very few neurons. Most of these neurons are those that have kind of been, the hypothesis of migrating up through into the cortex that kind of get stuck there. So we're really excited to understand more about oligodendrocytes at the transcriptional level, because typically neuroscientists focus on neurons, and to determine whether there are different types, subtypes of oligodendrocytes. And so far, you know, if you're a splitter, bioinformatically, you will find subtypes, but others in the field, particularly those who have been studying multiple sclerosis at the cell type level, also believe there are subtypes of oligodendrocytes. So we're looking at this not just over development and evolution, we also have some disease data sets. That data is not yet prime time to show, but in autism, so postmortem, we have 120 multiome samples that we're writing up right now. But in addition to that, postmortem tissues, this idea of trying to understand functionally how the OPCs and neurons might interact is something that is impossible to do using postmortem tissues, right? So we have to use other models. And so now in the lab, Wanjun Hong, I didn't put a picture up, he's a postdoc in the lab that moved to UCLA, is using iPSC-derived models in the room, in the back, to try and understand how neurons and oligodendrocytes between humans and non-human primates may interact and differentiate. And so these are just some preliminary data that Wanjun generated for a number of grants that we've submitted over the past few months while we're waiting for the lab to get set up. And I know it's a lot all at once, but here I'll just walk you through it a bit. So on the bottom are cells, human cells. On the top are chimpanzees, and we have multiple lines of both. And staining here for OLIG2, which captures the whole oligodendrocyte lineage, PDGFRA, which is an immature oligodendrocyte marker, and O4, which is a mature marker. And hopefully we can appreciate, even from the black and white, there's way more PDGFRA signal here in the human compared to chimpanzee. And if he quantifies this, basically what he finds is this increase in immature signal, decrease in mature signal in this 2D cell culture model. So we've essentially recapitulated what we found in the post-mortem tissue in this cell model. And this was challenging to do because Wanjun had to actually develop a protocol to make essentially pure oligodendrocyte culture. And so now what he's doing is it's seeding these cells with specific cortical layer specific neurons, either upper layer or lower layer neurons, to determine whether the presence of a human or a chimpanzee neuron, swap it with a human or a chimpanzee OPC, can affect gene expression, of course, because we love genomics, and then also activity. And we can measure the activity by putting these on a multi-electrode array. You're going to see a lot more MEA data coming up. So we're very excited about this. And so maybe if I ever get invited back to this in a few years, we'll have this, well, it should be published by then, results. So this is how we've been following up. And I never thought that the lab would move into, I'd say like 50% of the lab working on white matter and oligodendrocytes, but you follow the data, right? And that's what we've done. And I'm very excited about these results. So this is what the lab at UCLA will be doing, a lot of it. Okay, so everything I've showed you is basically, well, except for this, from post-mortem tissues for our comparative stuff. And I'm very interested in trying to understand human-specific signatures of gene expression, but with post-mortem and you're thinking about how, thinking about how cells may respond to activity, it's really hard to do. So I've always been driven by this question of, well, how can we be measuring these things in a living human? The dream would be that we can measure gene expression in a human like me while I'm walking and talking to you in some sort of like scanner or something, maybe not walking, but talking. So for a number of years now, we've been collaborating with a neurosurgeon back at my old institution, UT Southwestern, Brad Lega. And Brad's a very busy epilepsy surgeon. He's very interested in trying to understand the physiological aspects of memory. And unfortunately, a lot of patients who have epilepsy, medicines don't treat that. And so they have to undergo a number of kind of invasive measures, in this case, intracranial EEG, to map their activity prior to determining how they're going to do the surgery to remove the site of the foci of the epilepsy. So we're trying to figure out how can we intersect measures of gene expression with measures of brain activity specifically related to memory. That was Brad's interest. So we started with that. And these patients with epilepsy get the electrodes implanted, and then they undergo a memory task. They're shown words on a screen, then they're shown some math distractor task, and then they're asked to recall the words they saw on the screen while the neurosurgeon is recording all sorts of oscillations that are occurring in the brain. And so one of the first collaborations that we did, so two talented postdocs, was to intersect some postmortem, in this case, gene expression data with these intracranial EEG measures broken down by specific wavelengths of oscillation and ask, could there be a correspondence between gene expression and brain activity? And my lab and we were successful in this, and we also had some other studies where we intersected gene expression and measures of brain activity using fMRI. So this is reproducible. Multiple labs have done this. You can take these two populations, so postmortem, living patients, look at these two measures and find correspondence. And so I was glad that we could do that. I was excited we could make some insights. We could publish some papers together. But ultimately, there's a limitation, right? Living and postmortem. And I thought perhaps we could do better. And because Brad is such an active neurosurgeon and was game to do this, we were actually able to push the envelope further. And so these are just some schematics of the area of the brain that a neurosurgeon typically might have to remove to get to the center of the brain where the seizure often is, typically the hippocampus. So Brad is frequently removing what is called here the temporal pole or temporal lobotomy. And it's basically unaffected, quote unquote, normal tissue. This is not where the epilepsy is happening. Of course, the patient has had epilepsy probably for most of their life, but we're very selective in the tissue that we use moving forward for our studies. So the tissue has to be pathologically normal, no sclerosis, no heterotopia, and then we can proceed with using it, given the caveat that all of the patients have epilepsy. So we were able to develop this study where there are patients who had electrodes implanted. They underwent this memory testing physiology. They also had some imaging. Tissue was resected, the normal tissue, and then we could carry out genomics. In this case, we started with some bulk RNA-seq, and then you'll see single cell and single cell attack-seq, and then we could correlate all of these. Again, the patients have the same memory task, except we're limited now to using the physiological data from the temporal pole, which is the data the tissue that's being resected. And this was published a few years ago now. We ended up over the course of 5 or 6 years or more, 16 participants, which was kind of remarkable actually, for which we were able to get both gene expression data and these measures of physiology during the memory task. We also had some additional controls built in for bootstrapping and permutation analysis. And through all of this, we were able to identify genes associated with these various memory-relevant brain oscillations. And this is just the result of the multivariate analysis broken down by wavelength. We then further refined the list of genes correlated using Spearman's rank, and we were left with a list of about 300 genes that corresponded to a number of these different wavelengths. So very exciting, a list of human memory activity genes. And we wanted to know how specific was it, because we only had 16 patients. So we also had oscillation data from while they were doing the math task. We also had quantitative measures of their memory performance, and patients had to reach a certain bar to be included in the study. And we also had measures of cortical thickness, and we could correspond all of those to the gene expression, but none of those genes, except for one, overlapped with the memory relevant oscillations, suggesting this was very specific. Okay, so how could we go from 300 genes to a handful of genes that we could then study in the lab? So again, using WGCNA, as already mentioned, we could make modules of co-expression and overlap that with various lists of genes and identify this one module, WM12, that was significantly enriched for the wavelength where we saw the most genes, the delta oscillations. It also significantly overlapped with our prior 2018 study where we did bulk RNA-seq in independent populations. And this is just an illustration of the top 10 genes in this module. SHANK2 and SMAD3 were two that really rose to the top and caught our attention for a number of reasons, which you'll see later. And if you run a gene ontology, it's enriched for voltage-gated ion channel, things involved in neuronal function. Cool. If we do the same thing and overlap with GWAS and other data sets, remarkably, we don't see an overlap with epilepsy-relevant genes, but we do see overlap with some of these cognitive gene sets, which was really great, including schizophrenia, autism, bipolar disorder. So that was pretty remarkable. But we wanted to narrow down. Remember, this whole seminar is really about cell type-specific transcriptional networks. Well, what if we took single-cell RNA-seq now available at the time and apply that to the samples that were used in this study? And then we overlapped our modules of memory-relevant genes. What would we find? Well, we found, interestingly, that WM12 is significantly enriched, in particular for this lower layer, excitatory layer 4 to 6, keep that in mind, cell type. Very cool. What about the regulation? How do these genes hang together within this cell type? So now we could apply single-cell attack-seq to the data set and identify, here's all the different cell types, perhaps transcription factors that are regulating the genes involved in human memory. So if we do that and we look at WM12, one of the transcription factors that's significantly enriched is SMAD3. And we found, interestingly enough, motifs for SMAD3 upstream of SHANK2. Now, if you're paying attention, you remember SMAD3 and SHANK2 were in WM12 co-expression module. So right now we have two genes of high relevance to us with respect to human memory performance. And what we're doing in the lab, which I don't have a slide for, is we're trying to manipulate those genes in a human slice culture that you're going to see at the very end. Okay, so what we were able to do was to take this human memory relevant behavior. We were able to identify genes specifically correlated with just one type of, for example, oscillatory memory relevant behavior, identified the cell types, a lower layer cell type, and then identify transcription factors that could be regulating expression of human memory relevant genes in this cell type. Really amazing. So we're doing a lot with these two genes, but we don't want to stop there. Brad and I and the group are always trying to push the limits. So we thought, well, Memory is great. We're really interested in it. What other neurosurgical procedure has there been, like, not enough information on the molecular genetic mechanisms for which you may want to develop some sort of therapy in the future? So deep brain stimulation has been pretty effective for a number of neurodegenerative disorders. It's also become effective for restoring memory to a certain degree. So what are the cell types that are being affected when the neurosurgeon puts the electrode in and gets a positive response? Well, there's so many theories. Some papers say neurons, some say astrocytes, some say oligodendrocytes. We don't know. They're just guesses. So the team, so this is Haley Moore, who's an MD PhD. Well, she got her PhD this summer. Now she's back to the clinic. Anne Freeland, a graduate student who's in the audience, who moved with me, and Mantra, a postdoc in the lab who stayed at UT Southwestern. We have a number of hypotheses that we want to test using this system that I'll show you we've developed in the lab. What are the cell types that are important for neuronal or deep brain stimulation? How are the circuits changed? How do the gene networks change? How could we possibly ever even try and study this using a human system? So again, going back to this idea of neurosurgeons acquire a lot of temporal pole cortex, or at least Brad does over the course of his busy neurosurgical practice. And a number of labs over the years have shown that one could take this tissue and keep it alive in culture. And what we have been able to do, or the team has been able to do, is not only keep this tissue alive to do the gene manipulations like SHANK2 and SMAD3 that we're actively working on, but measure how stimulation, in this case, electrical stimulation, trying to mimic deep brain stimulation, may affect the genomic properties of this tissue. So here's the overall design. This is a bioarchive paper, Haley's dissertation that we're trying to get sent out. So this is all unpublished. Well, it's a preprint, but it hasn't undergone peer review yet. So we had 11 subjects for which cortical slices were generated, and then they were put on this multi-electrode array. I'm going to show you, it's a Maxwell. They're recorded, they're stimulated electrically using these paradigms, only important for the neuroscientists. And then we're recording the activity. And then importantly for the genomics group, we're collecting the tissue and it undergoes true multiome single nucleus RNA-seq and ATAC-seq. And here's just an example. I don't have the video, Haley usually shows, of kind of average firing rate. There are thousands of electrode on this Maxwell MEA. And the beauty of it is that the cortical thickness of a human cortex, it fits perfectly on this array. So we get the entire cortical thickness from the P, well, we remove the P, layer 1 all the way through layer 6 of the cortex with some of the underlying white matter. So it's a perfect fit. So Haley and the team have done this stimulation paradigm for a number of samples, as I showed to you. And one of the first things we wanted to know is, well, what are the cells that are responding? And the MEA is doing like a local field potential. It's measuring, it's not patching onto cell like some people in the room may know. So we don't know exactly what cell we're getting, but this may look familiar to you. It's kind of like, it is a UMAP, but of the physiological properties of the cells. And based on that, we can generate clusters, putting all the cells together. There's a lot of data cleanup along the way, which you can ask the team afterwards. And then we can make a pretty informed guess that these clusters are likely excitatory pyramidal neurons, and these clusters of physiological signature are they are the interneuron, the inhibitory neurons. And then we can ask which among these clusters of cells are responding to the chemical stimulation. Here's the example where we're showing 4 of these major clusters are responding to the stimulation, and 3 out of 4 of these are the putative pyramidal excitatory neurons. So it seems like excitatory neurons in this culture system respond robustly to this DBS-like stimulation. And then we can ask, well, okay, now we know the cell types roughly from the physiology. What are the gene expression signatures, and in which cell types are we seeing changes in gene expression when we took the tissue, dissociated it, and did the multiome? This is just an example. This is true multiome integrated, only keeping nuclei that have good RNA and attack signature. Reclustering the excitatory neurons, because the physiology pointed to us that perhaps the excitatory neurons are the ones that respond the most. Here are just the marker genes. And running a differential expression, stimulated, non-stimulated samples. What you can hopefully appreciate is we get genes going up and down, but this layer 4 neuron, IT is intratelencephalic, have the majority of, and actually layer 5 too, genes changing that are upregulated in response to stimulation. If you remember from the memory example, the layer 4 to 6 neurons were also the cells that seem to be really responsive in the memory relevant activity. So this is pretty exciting. And then from the literature, and this is really for the neuroscientists in the room, there's this whole literature of how neurons respond in a short, medium, and long-term way to activity, these activity response genes. And we found a significant enrichment of those in the cells that are responding to this chemical stimulation. Some you may recognize, FOS, ARC, EGR1, so things that make sense. But not only do we have the known players, now we have additional genes in human brain tissue that are responding to activity. So we have a lot of really cool genes to go after. Then we can use the attack data to build gene regulatory networks. The bioinformatics folks probably love this. Using Scenic Plus, there are lots of tools out there. This is just one that we used. And this is a really busy heat map, but it's basically highlighting here the regulons, if you will, that are changing in response to stimulation in a cell type specific manner. And again, highly enriched in this layer for these lower layer neurons. And in red are some known activity response genes. And you can build these beautiful visualizations of the specific genes and who they're connected to. And also looking at the accessibility of the chromatin as well as the gene regulation. So I'll just kind of show that to you, but I won't go into detail on it. Okay, the final piece, I know I'm running out of time. Brad also had a number of patients in which he does stimulation in the intact individual. So he needs to, these are patients who have a different form of epilepsy. He needs to map where it is before he removes it. It's not the temporal pole, other areas. And so, and then that tissue needs to get removed. So we were able to get from 9 individuals tissue that had been stimulated in vivo or adjacent tissue far enough away from the stimulation zone that's considered unstimulated. And then we carried out the RNA sequencing. So here's just the excitatory neurons UMAP. Now you can see here's the differential up and down with stimulation. The pattern is a little bit different. So we don't quite get the same result. Different brain region, slightly different stimulation parameters. We still get some of the same immediate early genes. Here's NPAS4, FOS, EGR2, but we get some other additional signatures here. But what was also really nice is we did this kind of like hypergeometric overlap of the in vivo and the MEA, the ex vivo slice culture genes going, because comparing gene to gene is not always the best way, but you want to look at the overall transcriptional signature. And we found that in the lower layer 3 cells, as well as the layer 5 cells, that there was this significant overlap of genes going in the same direction, both up in vivo and up on the MEA slice culture. So now we can say that even though we have differences, importantly, we also have similarities, suggesting that whatever we're doing in this ex vivo slice culture is a recapitulation of what's happening, hopefully, in a manner of deep brain stimulation or other forms of stimulation. As you can see, the number of genes itself is low, but interesting because we get some of these immediate early genes. Okay, and this is just a summary slide that Haley had made, which is really nice. That with stimulation, this is for physiologists, we see changes in activation, plasticity, and drift, which I won't get into. And that we really found this focus on these lower layer 4 neurons in terms of assembly as a physiology term, but also activity response genes, differentially expressed genes, synaptic genes, and the gene regulatory network. So we're able to hone in on a specific cell type using this approach. Okay, that's all I have to say. Thank you for your attention. I want to thank my lab, especially the ones that moved with me and are in the audience, and funding, and happy to take questions.